Great, thank you. So today I'll be sharing with you some results of a fairly long-term study that my research team has been doing, looking at variation among lake trout strains stocked in Lake Huron. There have been many people over the years contributing to this project. So the work began back in 2014, and so there's been a lot of people that have helped with this work. The work has taken place at the Codrington Research Hatchery, and so all of the staff at the hatchery have been obviously very invaluable for helping keep our fish healthy. As well, my research team has been visiting the hatchery at times weekly over the course of over 10 years to take some of the measurements on the fish that I'm going to share with you today. Chris Wilson helped a lot with getting this project off the ground. There's been also staff at a number of other hatcheries that contributed fish for this project. And then funding for the work has come through the Canada-Ontario Agreement on Great Lakes Water Quality and Ecosystem Health. And it's been great to have this funding sort of year over year to keep this project going. Okay, so with the collapse of lake trout populations that occurred throughout the Great Lakes, this prompted agencies to begin rehabilitation programs for lake trout. So in Lake Huron, we have fish community objectives related to restoring lake trout as the native top predator in the lake, as well as to restore self-sustaining populations of lake trout and to support commercial fisheries. As part of these restoration programs, agencies have been stocking a number of different strains of lake trout over the years, and that's really kind of the focus of the study that I'm going to share with you today. This graph up here is showing you sort of the history of 6 strains that have been stocked in Lake Huron. There's been other strains stocked, but these 6 strains are the ones that I'm going to focus on for my study. And as you can see, stocking sort of began in 1990 for these strains, and there's been sort of different numbers over time. So this represents the numbers of these strains stocked throughout Lake Huron. The numbers sort of look different according to the basin. The strains that we'll be looking at today include Big Sound and Iroquois Bay. So these are fish that are represent remnant populations from Lake Huron. So these are native strains to Lake Huron. Lake Manitou strain. So these are fish originating from a small lake on Manitoulin Island in Lake Huron. So these are sort of close to being a native strain to Lake Huron. Seneca Lake strain, of course, this is a very prominent strain in the lake currently, and so this originates from the Finger Lakes region in New York State. As well, we have a couple of Lake Superior strains. This includes Michipicoten Island and Slate Island. And I should mention sort of the reason for, or the rationale given for stocking these different strains is to increase the life history and genetic diversity of fish being stocked into the lake, with the hope sort of that one of these strains at least would take hold in the lake. And when these stocking programs, began, there weren't a lot of remnant populations from Lake Huron from which to draw. So we just had the Big Sound and Iroquois Bay. So this is what prompted agencies to sort of look elsewhere to bring in some of the Lake Superior strains and the Seneca Lake strain as well. Agencies have been monitoring the success of these different strains in Lake Huron to look at how well these strains are performing in the wild. So this is data given to me by Wendy Lee Stott, and the information can be found within a report that was prepared for the Lake Huron Committee. And what this shows is the contribution of these different strains to wild reproduction. So basically, we're doing some genetic analysis of wild-caught fish in order to look at the origin of the parents of these fish, essentially. And so these are just showing some results for the Main Basin, North Channel, and Georgian Bay. And the take-home message here is that we see a couple of the strains seem to be doing quite well. So this includes the Seneca Lake strain and the Lake Manitou strain. And this is when you look at the numbers of these fish in the wild versus how many are stocked, you see that these strains are doing disproportionately well compared to the actual numbers that are being stocked. On the flip side, we have a few strains that are doing disproportionately poor compared to the numbers stocked. So this includes, for example, the Slate Island strain, as well as the Michipicoten strain. And so the intent of this study that I'm sharing with you today is to understand the extent to which life history differences among these strains may be contributing to some of the differences that we see in the success of these strains in the wild. So the fitness of strains in the wild, of course, is a function of the numbers of fish of that strain that are surviving and reproducing. And this is a function of the genes of those fish. So these represent or are coded by like sort of intrinsic characteristics like life history traits, things like growth, reproduction, maturity. We have behavioral traits that could vary among these strains and have a genetic basis. And there could be physiological differences among these strains that have a genetic basis. Of course, how well these strains do is a function also of the environment. So you want sort of a match between the life history traits, for example, and the environment in which these fish are stocked. So just to give you an example, a lot of people have hypothesized, and there's a little bit of evidence for this, that the Seneca Lake strain, for example, are particularly good at evading sea lamprey predation. And this is because of the depths at which they inhabit. So this could be, a behavioral trait. It could have a genetic basis, and it could explain sort of some of that disproportionate success that we see of the Seneca Lake strain. Now, despite our sort of fairly long history of stocking these different strains in the Great Lakes, we actually have very little published work on how the life history characteristics of these strains actually differ. And so for this study that I'm sharing with you today, what we are doing is comparing the life history traits of these 6 strains in a common garden environment. And this is allowing us to look at these sort of intrinsic differences between the strains. And the underlying purpose of this is to understand the differences between the strains and to hopefully learn more about why some strains are performing better in the wild. We also looked at some physiological differences between the strains and how they performed at different temperatures, but I'm not going to share those results with you. So as I mentioned, the 6 strains that we looked at are up there. So this work was conducted at the Codrington Research Hatchery. So this is a research hatchery that's run by the Aquatic Research Section of M&R, where I work, and it allows us the opportunity to do these kinds of experiments. So for this study, we raised fish and basically followed them through from the egg stage to 2 years post-maturity. So the work began in 2014, and we just culled our last 6 fish that were remaining in this study just about a month or so ago. And as I mentioned earlier, we checked these fish on a regular basis, and we measured things like growth, survival, fecundity, age and size at maturity, those sorts of things. The Codrington Research Hatchery is a spring-fed flow-through system. So basically, the water temperature is kind of like the ambient temperature of where the water is coming from outside of the hatchery. Yeah, I mean, this is pretty basic stuff. We did follow fish as families in the beginning and then eventually track them as individuals. Okay, so we did look at the early life survival. So now I'm going to share with you basically just some of the differences that we saw amongst these different strains. It probably is a little bit hard to see, but there's letters above the dots there, and those tell you whether there are significant differences among the different strains, but I'm just going to basically summarize each of these differences. So what we saw very quickly was that the Manitou strain consistently had the highest survival amongst our different strains. So you can see that up there, up there, up there, and up there. And then the other very obvious result that we had was that the Michipicoten strain had very low survival. So you can see, for example, down here, during the incubation period, we had actually quite high mortality of this strain. And so we actually did try to get some additional Michipicoten fish, and they also, the offspring of those fish also had very high early mortality. We looked at growth both before maturity and after maturity. And the reason for doing this is because, before maturation, all of your growth is being invested, or all of your energy is being invested into growth, whereas after maturity or in the year leading up to maturity, some of your energy is now going into producing gametes. And so we looked at growth differently for these different life stages. And here again, we saw some pretty clear differences among the strains. Essentially, we see here that our Michipicoten strain had the slowest growth rates, whereas our Big Sound fish had the fastest growth rates. In terms of post-maturity growth rates, we did see differences between the sexes in the L infinity value. For females, Big Sound and Michipicoten fish had the highest L infinity. So these fish reached the largest sizes on average, whereas Manitou females had the smallest L infinity. So you can see the Big Sound fish up there, and then the Manitous, that their females had the lowest L infinity. Maturation. So this is showing both the size at maturation as well as the age at maturation for the different strains, and it's separated into males and females. So females are in the orange and males are in the black. And this was just determined by visual inspection of the gonads and whether gametes were released. So during the spawning season, we would go on a weekly basis to check each fish to see if they were mature. And so what we found here is kind of summarized up at the top. So we see that Manitou and Seneca fish had the earliest age at maturation and the smallest size at maturation, whereas Big Sound and Michipicoten fish had the highest size at maturation and the oldest age at maturation. And so you can see kind of how these fish fall along this, sort of going from youngest and smallest to oldest and largest. We also found that the Seneca and Manitou strains had a much narrow band of maturation. So most fish were maturing at that 3-4 years of age, whereas the other strains were more spread out. So there were some individuals reproducing earlier in life and then others sort of like following up later. So they had much more spread. In terms of fecundity, we also saw some very clear differences between the strains. The Slate Island fish had the lowest fecundity when you look at it relative to their body weight, as well as just the lowest absolute fecundity, whereas Iroquois Bay had the highest absolute fecundity and the highest relative fecundity. We also found that the Slate Island fish, they didn't have very high fecundity, and they were also just very sort of tough to spawn, if you will. So the eggs were not released very easily from these fish. Okay, so just to summarize some of our key findings. So we found that the Manitou, Seneca, and Iroquois strains had generally similar life histories, and that our prediction is that these life histories would likely give them a reproductive advantage in the wild, sort of all else being equal. So we see this higher early life survival, and this was especially true for the Manitou strain. So this, carries with it some fitness advantages. We saw fast juvenile growth, especially for the Iroquois Bay strain. And then we saw early age and small size at maturation for these strains as well. And this is what we think could really give these strains some advantage, right? So the time between when these fish are stocked and when they reach their first age and size at maturity is shorter for these strains. So that could give them that sort of advantage in terms of performance in the wild and being able to contribute to the next generation. We also saw high fecundity, relatively speaking, for these strains. On the flip side, those Lake Superior strains, so Michipicoten and Slate, may have more of a life history disadvantage. They have these kind of slower life histories, if you will. And the interesting thing there, of course, is that this does to some extent reflect what we see when we look at the contribution of these strains to wild reproduction, sort of coming from that graph that I showed you at the beginning, looking at the genetic analysis. I should mention we also found some interesting differences in the metabolism of these strains. And we did publish some results on that one. So you can check out that paper. Don't really have time to get into it today. So what we're now doing, so this study in terms of following through these fish is wrapped up now. And now what we're doing is we're building a life history model where we're basically putting all of that information that we just found, all those different parameters. And we've already built the model. And what this is going to allow us to do is basically to test that prediction. You know, do we see some advantage for some of these strains considering their life history traits above others? And so just on the bottom here, you can see just like some example results that we could have. So we put these fish into the model and then basically sort of let the model run, and then you can look at how well these different strains do in terms of their population numbers and their contribution to reproduction. So that's what we're sort of shifting to looking at now, and the idea is to get feedback from the Lake Huron Technical Committee, sort of for helping to build this model and making it as realistic as we can. And with that, I'll take any questions. This is my daughter who got to come for Take Your Kids to Work Day and got to sample some of the fish with us. So that was a pretty awesome experience.